Here is the villi, right? Multiple villus. This is, I'm going to actually just do that one, is a microvillus. If we have more than one, there we go. This is the lumen of our intestine, where chyme, which becomes more and more digested as we go through, that's where that's located. We've got interstitial fluid, and then we've got a capillary. Our goal is to get stuff, let's just put nutrients for now, from the lumen into the capillary. In order to do that, we have to go through the epithelial cell. These are here, this little thing right there is a tight junction. You remember that epithelial cells are linked by tight junctions. It was a feature we talked about in the fall. So closely linked, stuff cannot go in between, right? We can't go in between cells. We have to go transcytosis through the cells. So this is going to be our route of absorption. Two more things to label here. This is going to be our apical surface. And these are terms we learned when we talked about the features of epithelial tissues. They have an apical surface because they have an outside surface. That's what the lumen is. The apical surface of your skin cells is the stuff that's facing the air, covered in keratin in that case. This is our basal or basolateral surface. Also called membrane. Okay, so this surface here is a membrane. And actually, I want you to think of it as a membrane because we're going to have to think about going through that membrane. Let's cross that A out. So absorption can occur through several different mechanisms, right? How do we get things to do this? It's transport mechanisms, which I love. So we're going to be referring to active transport. What does this mean? This means it uses ATP. That's what that means. It's a pump. ATP pumps. Diffusion. Diffusion by nature is passive. And it can only occur for hydrophobic substances. Right? That's going down an electrochemical gradient as well. Facilitated diffusion. What does this mean? This means we're still doing diffusion, but we're not just going through the membrane. We have a facilitator, a protein channel or carrier. This is through the membrane. Phospholipid bilayer, I'm actually going to say. Carrier. Secondary active transport. This is a type of co-transport. We're definitely going to review this. Does not directly use ATP, relies on gradients that were set up by ATP pumps. And endocytosis. We won't actually use this one in the methods we go through, the mechanisms, but it is a possibility. Endocytosis and then exocytosis. This happens so then depending on what that nutrient is, it's going to use different processes. So for example, you already know lipids are able to diffuse through the plasma membrane. This means they are using what process here? Passive diffusion, not facilitated diffusion, because they don't need a protein to carry them through. They can diffuse right across that phospholipid bilayer. We're not going to talk about fats more than this. So this is it right here. You've seen this before. Bile salts emulsify the fats, allow lipase to access them. Pancreatic lipase chemically breaks down the triglycerides into smaller chemicals, fatty acids, for example, which then can diffuse through the phospholipid bilayer. The fats are the one exception to this capillary being the goal. This capillary would go to the hepatic portal vein. Fats are going to actually enter this lacteal to go to the lymphatic system. Eventually, will also enter circulation, right? So I'm going to go through proteins and carbohydrates in their mechanisms of absorption rates. You can tell this is a piece of toast, right? Toast contains starch. Starch is a polysaccharide. The same process would apply if you ate other polysaccharides and could possibly spell them. I can't spell this right now. Saccharide. Starch is going to be broken down into disaccharides. In the process of that, it doesn't just go from like long chain to disaccharides. You're becoming oligosaccharides in between. But to get from starch or other polysaccharide to disaccharides, it's amylase that does that. Salivary amylase is one location, and then pancreatic amylase. So depending on how much you chew, you may do more of this process in your mouth versus in your duodenum. You want to give your pancreas a break, maybe chew for longer. I don't know if that's a thing. I think your pancreas will still produce amylase. So salivary amylase and pancreatic amylase break down starch, glycogen, other polysaccharides that you're eating into these oligo and then disaccharides. To go from disaccharides to monosaccharides, which is what we need to absorb, we need to have brush border enzymes. So let me go to monosaccharides. Am I spelling it the same way at all? I think it's supposed to be A in there. I'm not going to test you on your spelling of saccharides, okay? Monosaccharides that are absorbed include galactose, glucose, fructose. Those are common monosaccharides. Just to give some context, let me add a few disaccharides. So lactose and sucrose are probably ones you've heard of. You also could ingest lactose or sucrose just straight up, right? And not have to have your salivary amylase do anything. You also can eat glucose and fructose. But this is if we're starting with the polysaccharide, with this piece of toast here. So the enzymes that do this are brush border enzymes. Where are brush border enzymes located? In the brush border of your enterocytes. And I've got a picture of this in a moment. So this is in the small intestine. There's all types because we need to break down a lot of different types of disaccharides. So lactase, maltase, sucrase, dextranase, etc. Let's look at this with the picture. Here is our starch molecule. Let's assume that our saliva didn't do much, or maybe we swallowed it without chewing, swallowed a piece of toast. Here's our starch. It's broken up by pancreatic amylase to form these disaccharides. Disaccharides still can't be taken into the enterocytes, the absorptive cells. They need to be broken down. That occurs via these brush border enzymes. So we've got 1, 2, now they can be absorbed. Absorption is this whole shebang here. This guy is also going to be important, but this is the actual process of absorption. We're getting glucose, let's just call it, from out here into the capillary. Let's look at how we do this. Hopefully you're realizing you know this already, and I've taught you it so many times. So this might look familiar. How does glucose move from the lumen of the intestine to the ECF, ultimately then to the bloodstream? Let's add bloodstream here. Capillary. ECF, that's the same thing as ISF, right? Interstitial fluid, extracellular fluid. Interstitial refers to around the tissues. Extracellular refers to outside of the cell, anything outside of the cell. Capillary fluid would actually be a type of ECF. Okay, the anatomy here you've seen, right? Apical surface, basal or basolateral surface, tight junctions between epithelial cells, so we cannot move this way. We've got to go through the cell. There is high glucose inside the cell, lower glucose in the lumen. If you've just eaten a large amount of glucose, this might slightly be different, but basically this is going to stay real high. It's all relative. This is going to be low here, partially because it's being carried away into the blood constantly. So how do we do this? First of all, we need some proteins. We need 3 proteins. We need to establish a sodium gradient. We're going to have secondary active transport here, right? We need to have a sodium. Well, first of all, we just need this protein always. All of our cells have high sodium outside of them and low, relatively low sodium inside. This is established by our sodium potassium ATP pump that is present on all cells. This pump establishes this gradient. So given that, we can now use that gradient to co-transport sodium with glucose into the cell. Why couldn't glucose just move without that co-transporter? It was not down its gradient. There's high glucose inside the cell already. It cannot diffuse even with facilitated diffusion into the cell. It can't, it's not going to go that way. There are not pumps for large molecules like glucose, so this co-transporter is used to bring glucose in along with sodium. Sodium's going down its gradient. Once glucose is in here, so glucose is moving in, glucose, lots of glucose coming in. There's lots of these co-transporters. It can then diffuse via facilitated diffusion out a glucose transporter on the basal surface. So apical surface, we've got the co-transporter. Basal surface, we've got the glucose transporter. We don't need to use sodium here because this is down its gradient. So let's label what these things are. Choose a different color here. Let's do this one. This is facilitated diffusion. This is active transport. This is co-transport and specifically secondary active transport. So we're eating protein. Let's say we eat a big chunk of steak, or maybe it's rice and beans. That's going to be broken down first by pepsin. Where is pepsin produced? In the stomach glands, activated by HCl. Those proteins are going to be broken down into polypeptides that are smaller than before. We then have another set of peptidases, pancreatic enzymes. So trypsin and chymotrypsin are the two that I ended up having on the learning outcomes. The other one is that carboxypeptidase. These are going to break down the polypeptides into small polypeptides / peptides. What's the difference between a polypeptide and a peptide? Some kind of, there's definitions of like how many it has to be. I think it's like 50 to be a polypeptide. I don't know. They're getting chopped down smaller, right? We need to get eventually though to what? Amino acids. Amino acids are the smallest unit of proteins, and this is what can be absorbed. Some di and tripeptides can actually be absorbed, but let's ignore that. Brush border enzymes are going to do this final step. So this is in the intestine, right? So there's aminopeptidase, dipeptidase, other names, because we're breaking down slightly different things here. The substrates are going to be slightly different. Now, transcytosis can occur a little bit, but we're actually mostly going to go through the cell again, just like we did with carbohydrates. This is what that looks like. Nothing new here. Look, amino acids broken down by pancreatic proteases. I'm going to add in here because we're in the intestine. In reality, some might have occurred in the stomach as well, right? But if we're in the intestine now, this is step one in the intestine, pancreatic proteases, followed by brush border enzymes to get to our amino acid. I don't think this is, look, that says amino acid carrier. So this would be an amino acid. We are going to use a different color. What do you think the name of this transporter is? The one I'm about to label here. What is it called? It is a sodium amino acid co-transporter. So co-transport usually uses sodium very commonly because we've got that sodium gradient. It is useful to use. It works. But it can be different things carried with our sodium. So amino acids can be used, can be brought in that way. Once they're in, that amino acid can go out an amino acid carrier, a lot like a glucose transporter, but it's for amino acids. This whole process relies on this ATP pump. So this again is secondary active transport.